Hello.
వస్తే తీసుకున్నా
Hi, everyone. Let's just wait 5 more minutes till everybody joins, then we are going to start it.
Hello. Hello. Hello.
Yeah, hello.
Good evening, madam.
Yeah, actually, we are waiting for a couple more minutes till everybody joins, then we are going to start, okay?
Okay, I'll be here only. Okay, madam.
Hi, everyone. I'm Suchismita Panda, President of Beautiful Mind Youth Mentoring Foundation. Sorry, my voice is completely dead today. Please bear with me. So welcome to this journal club. We have some amazing mentors who are going to guide you all through this 8 sessions journey. So I think you all know by now what Beautiful Minds do for the young generation. We are here to mentoring you guys. We are here to support you in any way we can. So this journey is about exposing you to the scientific research world, to talk about various journals, to talk about how to go by literature review and different fun experiments. So I will let Dr. Vibhuda Parasar start his presentation so that we don't be delayed, okay? Can you all please mute yourself? Everyone, please maintain the decorum. Okay, Dr. Vibhuda Parasar.
Oh, hi. Sorry, I'm trying to mute everyone. Yeah, I think everyone has muted.
And if anybody has any questions, please note it down. At the end, we will open the floor. You all can ask your stuff, okay? So that whoever is presenting, they don't have to be bothered by all those small questions, okay? Note it down, and then we can open the floor for the question and answer.
Hi, good morning and good afternoon to people from different parts of the world. Today we will talk about scientific research. More broadly, we will not discuss any specific data or like in the publications, but we'll talk more about scientific research in general, as this is just the first meeting of many that is there to come. Now, research, the scientific research is basically the systemic and creative actions taken to increase the knowledge about humans, culture, society, like everything. And when it comes to biological research, of course, it will be more about our body, our health, medicine, and things like that. Now, this is definitely different from of gaining knowledge by reading a book, because when you read a book, you definitely learn new things, but those are things already known to the mankind. So when you discover something that is not known to the mankind in general, that will be like a finding which is publishable, and that is called scientific research. Now, scientific research is a neutral, systematic, very planned, multiple step process that uses previously discovered facts to advance knowledge that does not exist in the literature. You see, I keep talking about this word called literature. So what is literature? Literature is basically this vast database of previously knowledge. It's basically, you can call it, is the encyclopedia of science. Now, as you know, everything is online. So all this knowledge is online now. So we look in the online database to look for any previously reported data based on our question of interest. And then once we have a question and once we look through literature, the type of question can be classified as observational or experimental. We will discuss those things later on. Now, all scientific investigation, they start with a specific research question and a formulation of a hypothesis to answer this question. Now, hypothesis should be clear, specific, and directly aimed towards answering the research question. A strong and a testable hypothesis is the fundamental part of scientific research. And then the next step is testing the hypothesis using a varieties of scientific methods to approve or disapprove it. Now, scientific method, as we talked about, should be neutral, objective, rational, and as a result, should be able to approve or disapprove the hypothesis. The entire research plan should include the procedure, the procedure to obtain data and evaluate the variables. Now, we will discuss what's a variable in a few slides. Now, all this data, once you have all the data, you have to ensure that this data can be analyzed. It should also include plans on the statistical analysis to be performed, because without statistics, you cannot say if the data, what you got, is proving or disproving the hypothesis. Let's say the hypothesis is okay, let's say we initially, when the COVID pandemic hit the entire world, in the early 2020. I am not talking about the earliest hit in China, but like the global pandemic when it hit the entire world around like February, January, February, mid-March. So people started to think, asked a ton of questions about COVID. What is COVID-19? How it is like, entering our body, how it is doing all the damages that it is doing, how to figure out a cure. So they started looking like, through literature. That's the very next that you do. But of course, it's a new disease. No one knew anything about it. But people knew that there's COVID-19, which is also called SARS-CoV-2. You may have heard this word SARS-CoV-2. That's because this COVID-19, this specific virus, shared a sequence similarity almost 80% with a previously reported SARS pandemic. I think it was in 2002 or something, SARS. Now, because it shared a lot of sequence similarity with SARS coronavirus, this was called SARS coronavirus 2, SARS-CoV-2, and then later it became COVID-19. They also found that, oh, hey, this COVID-19 also shares similarities with another type of coronavirus called MERS, which we also had MERS pandemic somewhere in the Middle East around 2008, 2007 or something. I don't remember exactly. Now, in the literature, they could not find anything about COVID-19 for obvious reason, but they found like in a lot of tons of data about the SARS epidemic and then the MERS epidemic. Now, starting those epidemic, they definitely sort of figured out some idea about how this coronavirus might behave and what the possible cure might be. So what they did, the next hypothesis was maybe the treatment that were used for the treatment of previously the SARS pandemic or the MERS pandemic might work for the COVID pandemic. So that's the first hypothesis they had. So what they did, they started the same routine for the COVID pandemic. And the second thing is when you're trying to find a new medication, and as you know, finding a new medicine is not a joke. It takes 10 to 11 years. So the best thing that you can do is look for medication approved for other diseases and see if they can also somehow magically cure COVID-19. Because that way, already those medicines, they have been tested in humans. People know they have minimal side effects. So if they work for COVID, it would not take another 11 years to approve some medication for COVID. So that was the next hypothesis, that maybe some of this, medication you see in CVS, Walgreens in the US, or in varieties of medicine stored in India, maybe some of these medicines could be effective for COVID-19. So in a varieties of in a hospital, both in the US, India, Europe, like basically all over the world, they started testing different medication in COVID patients. So what was the experiment? So a right experiment would have control samples. What it means is some patient would be given nothing, control sample, which can be a blank pill or just be a sugar pill. Now, control pill is called a placebo. So they are given a placebo treatment. And then in the drug category, they are given all this medication or this combination of medication. And then they would observe what happens to the control sample and what happens to the patients that were given this specific cocktail of drugs. Now, only if they see that, hey, in the control sample, the patients are not getting cured, but in the treatment sample, people are slowly doing better. Now, see, without the control sample, they would not have any data or conclusion about if the treatment sample is working or not. Now, once it works, they have to do it in not just in one patient, not two patients, but maybe tens or maybe hundreds of patients. Now they have to do in tens or hundreds of patients, so like hundreds of control, hundreds of treatment. Now then they will do statistics. Like statistically, is it significant? Now once it is statistically significant, then they will have the conclusion. Oh, hey, look, so this medication is working. And then they will publish the data. We will go through this specific component later on. And then once they publish the data, it is now available to the entire world. Remember I talked about this encyclopedia of biology, biological research. It's called PubMed. We will go to it like in a few slides. And once it is out, like, in this so-called encyclopedia, now everyone is aware of this cocktail. Now, based on now that becomes the existing knowledge now. Now, based on that existing knowledge, people will have new hypotheses, and then the cycle goes on. Now, this is the scientific method, and this is something we do every single day, not every single day, but like periodically. To discover new things in biology, but like in general. Now, as we talked about, a design of the research is perhaps the most critical part of any research. Without a proper design, proper plan, the entire project would just fall apart. You have to know exactly what you are going to do in any research project. How many patients, what treatment, what dose, when to give the dose. Do you want to give the patients the dose at one constant level or like in an increasing level? What's the dosage course? For how long do you want to do the treatment for? A ton of things basically, they have to be carefully designed to before starting any experiment. So it is very important to properly design a research, write everything like on a notebook or a notepad with every single aspect of the experiment has to be methodically written out. And then we talked about hypothesis, right? So hypothesis means, of course, this is exactly what it sounds like. You hypothesize, you guess something based on some previously established knowledge. It's not like just a wild guess. Oh, hey, look, I think when you have full moon, some people go crazy. No, it's not that kind of hypothesis. There has to be a scientific foundation behind it. Now, once you have a foundation, and then we start planning experiments like we discussed here, and then we then either prove or disprove hypothesis. Does X cause Y? Are X and Y are somehow related, even if X does not cause Y? Is there a correlation between X and Y? Like, even though X does not directly cause Y, when X goes up, Y goes down, or maybe when X goes up, Y goes up. So things like this is what we ask in the hypothesis. And then if you remember, we vaguely talked about two types of study in the beginning. One is called descriptive study. Second thing is called experimental study. In a descriptive study, you do not manipulate the experiment. You observe the experiment as it is. Let's say you are trying to study, like, for example, like, how people from, like, let's say Asia, like, Europe or maybe North America, how they respond to a specific type of food. So it's things like that where you just observe them. or you want to see, like, let's say in aging, when, like, someone is born until they die, in aging, how is the specific, like, blood component level changing. So you don't do an experiment on anyone, you just observe them as they are aging slowly. These are called descriptive study. This is more of a common practice in almost all the non-invasive human study. And then there is experimental study. This is what we do sometimes on humans when we do drug designing, but mostly on basically what we call model organism, which can be like, mouse, rat, donkey, horse, can bacteria, viruses, parasites, earthworm, Drosophila, like, fruit flies. It can be anything, because we can, of course, under certain guidance and rule book, we can manipulate the system to some extent. And then, like, we also, like we discussed, when we had COVID, we had to test different drugs. So of course we had to manipulate, like, what they are going to consume. So some of them will consume placebo or maybe just a sugar pill, and some of them will consume the real drug that we are testing. Now, so we have control sample, we have a manipulation sample, or which is also called a treatment sample. So we have all these two types of samples in an experimental study where we manipulate one group specifically. And an ideal study would basically randomly assign subjects, or basically, in for case of drug treatment, people to different groups. For example, if you watch, like, let's say, if you don't have experience with medicine or how the drug trial works, you may have watched like, few TV series on Netflix here and there. When people sign up for any drug trial, they don't know if they will be placed in control group or a manipulation group. They don't know that. That's called random assignment. And next we'll talk about variable. Now, variable in science is, I think it's very important to understand. Variable is something that we change, or variable is something that that is something the research is based on. Now, for example, you want to understand, like, how asthma, like, it works. So you basically look at if weight has any effect on this, if height has any effect on this, if all these varieties of factors have any effect on this. These factors, they are your independent variable. And the final effect, that's the dependent variable. So, but when you are doing a drug treatment, the specific drug dose, the dosage time, the dosage duration, and then the increasing or constant level of dosage, all these things, they would be independent variable. And the dependent variable is basically how the patient is responding or behaving with respect to the given dose. And then we have to see if it works or not. We have to plot the data, we have to do statistics and all those things. So, like, for example, let's say the independent variable is education level and the dependent is income level. When you plot the data, you might see a positive correlation. Same thing, exercise versus weight. The more you exercise, the more weight you lose. So you might see a negative correlation. And there are, like, more examples here. But basically you have an idea of cause and effect. Now, the cause is the independent variable, which is always plotted in the x-axis, with some exception, but it's in most of the cases is plotted in the x-axis, and the dependent variable, something we are observing, is plotted in the y-axis. So at the end of the day, it looks like y is a function of x. So dependent variable, if it is a function of independent variable or not. One more thing that you may do or may not do, these are only basically done in very high-end setting, like under a lot of security and everything, is the blinding experiment in drug trials. So if you know who got the placebo and who got the treatment, that's called single blind experiment. And if you don't know as a scientist who got what, and then that's called double blinded experiment. So basically you don't know who got what, but someone else knows. Someone else knows who is not involved in the research. Someone else knows which individual got, like, which treatment, placebo versus the drug treatment. So your job is to basically get data from individual people without knowing who is control, who is treatment. And then you analyze the data and then you conclude, oh, hey, look, this patient is doing much better than this patient. And then the other guy who is not involved in the research will tell you, oh yeah, this one got drug, this one got placebo. So that's called a double-blinded experiment. Then when we analyze the data, we have to think about precision, accuracy, and then error. Now, let's say you are testing a new drug, and then the new drug is supposed to lower your cholesterol level. For example, it doesn't have to be a COVID drug. Like, and then what you see, like, if this is your desired cholesterol level, and it's definitely like, around it, but it's still very far. It's not hitting the bullseye. It's not exactly lowering it to a desirable level. So of course, this cannot be a drug target. Let's say a second drug target it's basically, it's definitely, it's like, lowering it to something, but it's not to the desired level. It's lowering maybe slightly, but it's consistently, it is lowering slightly. So this cannot be like, but a desirable drug candidate. And then you have the actual drug candidate where you have your desired level, but also desired accuracy. So every single patient that got the treatment had their cholesterol level basically go down. And then, as we talked about, once we do all this, like, painstaking process of designing the experiment, collecting data, plotting the data, doing statistics, figuring out if this is the best solution or not, and then we make it available to the encyclopedia. And there are, like, certain journals. This is data from 2012. It's very old. Now, these are like some of those famous scientific and medical journals where, like, we try to publish all the time. And then majority ones that you may have to remember is we have varieties of journals from Cell Press. We have Cell, we have Cancer Cell, Cell Stem Cell, varieties of things from Cell Press. And then we have Nature Press, Nature Materials, Nature Genetics, Nature Nanotech, like, Nature Immunology, Nature Cell Biology, Nature Physics, Nature Neuroscience. And then we have Science. We have science, and you don't see other things: science immunology, science translational medicine, science this, science that. And then we have Lancet. We also have varieties of Lancet journals: Lancet Oncology. There has to be one Lancet. It's very old, 2011. So, and then you have New England Journal of Medicine, NJEM. That's also, it's not shown here, but it used to be in this area in 2011. Right now, and then you have JAMA, Journal of American Medical Association. They also publish a ton of highly cited papers. Now, what is this plot? Now, this plot is basically what we call impact factor. Now, there have been debates, like, for decades to if this is like, an appropriate parameter to assign to a journal or not. But, like, debates aside, it has been so far widely accepted, and pretty much everyone who is doing research, they're aware of these numbers. And this is how we decide where to publish in majority of the cases. So what is this? It's basically how many times a specific how many times on an average a publication in a specific journal gets cited by other people. So, like, for example, in 2011, an average paper from Cell was cited 35 times a year. So 35 different people, different groups across the world, they basically did further research based on the finding published in that report in Cell. That's like an average figure. So of course, some paper will get cited more times, some will get less times, but this is sort of an average profile of that journal. So this is sort of just to give you an idea of like, which are the good journals or the highly cited journals to refer to if you're trying to understand like, good science and good finding, trustworthy finding. In these journals, definitely the scientific rigor is much higher. The statistics are carefully evaluated, and the conclusions are carefully evaluated. And then, as I talked about this encyclopedia, this is published by the National Center for Biotechnology Information, popularly known as NCBI, in collaboration with NLM. And they publish basically, like, a varieties of database of everything for free to the broader scientific audience. Database of all the bacteria, database of all the viruses, database of all the sequence, all the taxonomy, all the protein, all the RNA, everything that we can think of, including all the publication. Now, this publication arm of NCBI is called PubMed. This is something either or maybe you will be using in the coming future very frequently. This is how it looks like. So PubMed, you type, like, your term of interest. Let's say I will type, well, this is a screenshot I cannot type here, but let's say I type COVID-19 and a specific drug to figure out, like, how many publications in the past so far have used COVID-19 and this specific drug, and they have mentioned this combination somewhere in the text. Now, for an example, we will just look at how, like, a paper looks when published in a scientific journal. This is one paper, like, in Cell, that we just talked about, having very highly cited, highly referred, highly respected journal. So this paper is about very interesting topic, interesting to people from India or from Indian origin, is So like when British invaded India, they tried to break India into multiple pieces. Their ideology was like divide and conquer, right? So they tried to push forward a theory called Aryan invasion theory, sort of like all this entire people, everyone from North India, they're somehow like descendant of uh, the invaders from Middle East, Iranian, Pashtunian, and then they somehow, and then basically pushed away the native Indians to southern part of India, and they are called Dravidian. So this is the famous Aryan invasion theory that they try to push forward, sort of to create a division between North and South India. And unfortunately, some of this textbook, they still teach these things, like colonial mentalism and everything, but we won't go into politics. We'll just discuss one of the many papers that came out in 2019 discrediting that theory. I picked this paper because it has a easy figures to understand. It has less complicated, I would say, statistics, or at least a graph to understand. So what happened here was they found, and this was done basically in collaboration with people from India and then Harvard. Sorry, I currently have COVID. So, and then, mm, um, yeah. So basically, what they found was they discovered an intact skeleton in Harappa, Mohenjo-daro, which is basically known as the oldest civilization in the world, and the civilization that basically gave rise to the entire population of India in due course of time. And their argument was all these people, they also came from the Iranian Pashtunian, that migration of people from ancient Middle East to India, and then they basically pushed away people from North India into South India. um, they basically sequenced. I will not, of course, discuss details, but using a combination of sequencing, machine learning, and everything, what they basically concluded is that people from Iranian ancestry, they did not migrate to the Indian peninsula, but in the Indian territory, but rather people from based on the gene sequence and other signatures, people from Mohenjo-daro, Harappa, they basically migrated to everywhere, to India, to even further west. And then over the course of time, I'm not showing any more figures, but over the course of time, it became normalized. There are even more complicated genetics papers out there now, I think from Stanford and MIT, Harvard. They sequenced everyone from North India to South India, suggesting we both had one common ancestor, not two different ancestors and everything, but we won't discuss this data today. But not all data looks they're simplified. Sometimes you have to do different types of plot, a complicated plot to make sense of complicated data. You may have to do a varieties of plot, varieties of statistical method to have a specific conclusion. So my point is figures and data in the paper can go anywhere from this simple looking to this complicated. So hopefully in due course of time, we'll try to understand different data, different figures, what they mean, how they arrived at specific conclusion, and so on and so forth. And then once we do all the experiment, now it's time to write something and then publish. So these are different components of research article. We have a title. The title has to be clear. It has to convey your message clearly. It has to be appealing to a broader scientific audience. And then we have an abstract. And in an abstract, we talk about the generic problem, and then we talk about the specific problem of why we are doing this study. We vaguely discuss the methods and then results and the conclusion. So it's basically a one short paragraph. If you read the abstract, you understand what's going on in the next 4, 5, 10, 12 pages. And then we go to the main body of the paper. We have one introduction where we talk about the general problem, and then the specific problem. And then we discuss methods and materials with a lot of details. Why? So that our goal is, if this finding is becoming public, so that other people can cite this and use this study to discover even further new things. This data, they have to be reproducible by other people. If other people cannot get the same data that we get or we got, so that's a problem then. So we have to we have to explain with a lot of details where we got the materials from, the exact materials, the exact method, the exact time, the exact dosage, everything, the exact regime, everything in great details. And then we discuss the figures. If we have any table to plot, we show any tables, we show data, and then we explain them, and then we discuss them. Then what's the broader significance of the data? We discuss the broader significance of the data, and then we also discuss, okay, hey, look, this is the conclusion, but this finding may have this limitation. So you have to also because you know more about your experiment and data than other people. So you have to be upfront about where your study could use basically let's say maybe new technology, or you wish you had let's say more diverse population. Let's say if you are testing a specific drug, you wish let's say you had patients from all over the world, things like that, broader limitation. And then future scope, where this finding can be helpful and everything, and where do you see the future, how do you anticipate the future finding to use this data and everything. And then there is this minor section called acknowledgement, but which is the most important one, where you thank everyone who definitely helped you, but not to an extent that they got a name in the publication. And then you thank all the people who basically funded the research, and then you have to declare competing interest. I will not go into all this boring details. At the end of the day, at the end, you have your references. This is again going back to which studies you refer to, you looked at before, planning, designing an experiment, which studies, which publication you refer to while analyzing your data, and so on and so forth. And the list of references is where these journals get the citations from. I think that's it for today. Yeah, if you have any questions.
Thank you, Dr. Parasar. It was interesting to understand your point of view, what you are doing, and you simplified everything. I just want to add few stuff. I was taking notes because it might be overwhelming for some. They might be thinking, what are the terminologies, this and that. So kids, it's very simple. It's very, very simple. So basically, when you start a research project to be published, there is multiple group of professionals behind the researcher. Okay, so it's a group of researcher, or a single person can do it, but there will be statistician, there will be various committees to approve, and then there will be this grant to conduct, to financially assist you to conduct that thing. So it's a lot of stuff go behind the scene. So first thing is, you have to ensure it is conducted well, abiding the rules and regulations. You cannot just wake up one morning and start collecting data. So there are rules for that. And the second thing is, all the literature, slowly you will understand, they are written in certain formats. Okay, it's just not layman English you can write and get approved. So there got to be MLA or APA. So those are different formats. If you pay attention to various literatures, you will see some of them are projecting the data in certain ways. So MLA has certain rules, APA has certain rules. All the I'm pretty sure Dr. Parasar has gone through all those hours and hours of study time, understanding what are those different formats while he was doing his doctorate. So those are very important part of writing a literature. Okay, and when you start it, the starting point of writing a literature is a hypothesis statement, what he mentioned. So the hypothesis statement is extremely important. That's, you take a good amount of time and try to understand what you're trying to find. So the hypothesis statement is something that will project your future work and what you are thinking in front of the committee. They will understand what that is. Then only they will grant you. Then only they will take you to the next level. Okay. So then you have to write this proposal to various granting committees and all. Then if human subjects are involved, then there are many guidelines, okay, to control the result, because they cannot be abused. So there are certain sensitive groups, like let's say minors, the mentally unstable, prisoners, women. So they have different kind of guidelines. So then you have to form a consent form. You have to go ahead, and once you get all those the permission to conduct the research, then you have to generate a consent form for various groups. You have to ask them their permission that whether you can use the data or not. You cannot just go ahead and use the data. Okay. So once you get the permission, then go the data collection. And every research article, see, we keep on referring to literature. So we call it literature, okay? So every literature, there's like 100 to 150 pages. They have various sections, as Dr. Parasar was saying. So we have an abstract section, we have the data collection, then you cite them properly, then there is references, there are like many stages. So all those APA and MLA, what I was saying, they actually teach you, guide you how to write that paper. Without that, it cannot be accepted. Okay, so now let's go to the data collection. So all the literatures are either qualitative, quantitative, case study, or mixed. Okay, so qualitative meaning when you are forming your hypothesis, when you are thinking in the future how I'm going to write that paper. Let's say, am I just collecting data without getting involved in those complicated mathematics stuff? So that's going to be pure qualitative. You are just collecting, let's say you are conducting a research around your community, where you're seeing how many kids are attending schools. Okay, for just for example, just a simple thing. so that you are not dealing with much complicated mathematical stuff. It's just a general research. You collect your data, you process it, and you publish it. But when it comes to quantitative is extremely complicated, where you go by all those statistics. Then you have to hire a statistician, or if unless you are extremely familiar with that stuff. Then the quantitative is extensively that simplifies and that projects all those data. What Dr. Parasar was showing, those graphs, and it can be simplified, it can be complicated. That comes under that quantitative research study. Then there is the case study. Case study is pure group. You follow a group, you have you try to find out the hypothesis, how that works on that group and all. You collect data and you publish it. And mixed when we'll go detail into those literature reviews. Again, literature reviews meaning there will be let's say we pick a group of subject, or let's say we are like my thing is oral cancer and liver tumor and aging. Let's say I'm targeting a particular population for oral cancer, let's say whoever are smokers and all. So I, instead of going into like just qualitative or just quantitative, I want to get a mixed result, because I want to study them and I want to gather their complicated data and I want to publish them in a different distinguished statistical manner. Then that's going to be a mixed one. That's a bit difficult, but most scientific literatures are probably mixed. Okay, so then how we say, like, there is an abstract, the body, then there's data. The main thing I want to point out here is a plagiarism. Okay, I'm not sure if you all are familiar with that term. For any research, even if you get your approval, this and that, you have to make sure the data are not plagiarized. Okay, so the citation, what she was saying, why citation is so important. Citation meaning you are giving credit to a previous published author's work. You cannot just pull their work and put it in your thing and do not credit them. That is wrong. Okay, so that's called plagiarism. Always, whenever you pick a literature, a published paper, you will see at the end there will be at least 100 of references where it's written properly where this data is drawn, and that's also gives you a authenticity. Like, this is not something you are pulling out of thin air and you are telling the world, see, this community is actually consisting of this, or this is their behavior. You cannot say that. You have to back that up with some previous research. That's called citation. Okay, so this is what I wanted to add. If you have any other questions, please go ahead. Then we will move to Dr. Bal. Dr. Bal can brief you of what he is planning to do. Do you have any questions? Someone has to start. This is the time. See, this is the journal club. We are here together. That's one thing I am insisting you all, please speak up. You all are professionals. You all are in a nice place in your life. Speak up. There is no judgment. There is nothing. You just speak up. Otherwise, it's very discouraging to talk, okay? You can ask that. Unmute yourself and ask that, please. Vaishnavi?
Hello.
Yeah.
Can you hear me?
Yeah, we can.
Yes, ma'am. I wanted to know if we can learn the data analysis part of the research.
That's Dr. Parasar. You're mute.
Sorry, like today or in the future?
I mean, I just wanted to know if it is possible. Like, I really wanted to know how to do the data analysis part. So it would be really great if you can teach us how to do
Yeah, I mean, definitely, like, we will discuss how the data is analyzed, but you have to understand different types of data. They have to be analyzed in different way.
It's the vastness. It's extremely
it's,
vast. It cannot be
it's
taught in just
huge.
yeah. So if you are interested, actually, If you're
Huh?
interested, we can start another one and talk about it. Okay, but we'll definitely touch that. We'll definitely touch how to go by it. Okay.
And then
Okay.
different types of data, they would require different types of statistical analysis. So there's tons of things like, small things here and there that you learn and you master with time. It's not definitely, it's not a one-day process.
Yeah.
It takes
I know that there are more sessions that are coming up, but it would be really great if you can teach us little by little in these sessions so that we are at least aware of it and talk about it.
Okay, we'll keep it in mind. Definitely, we'll keep it in mind. So there's an excellent suggestion, Vaishnavi. So kids, everybody, whatever you want, I mean, to add, whatever you want to say, we can definitely modify later. Okay.
Yeah.
Next, anyone else?
Thank you.
Okay, you're welcome. Anyone else?
Hello, ma'am.
Yeah.
I wanted to ask how important is the journal in which the paper is published and how important are the citations?
Oh, citation. I just told you. Plagiarism you can get sued. You can go to jail. Your license can be, I mean, it is extremely important. I cannot emphasize enough. You have to cite your work. You have to understand how to cite. There is also different format for that. You have to honor others' work. You cannot just write something, and you cannot, and that's also, it's just not about honoring. It's about uh, protecting the falsehood. Like anybody can say do some tall claim, and this is not backed up by some previous publications. So that's why it's very important that you go for citation. Okay.
No, ma'am, I'm talking about my paper. For example, if I've written a paper and I have to
Okay.
publish it in a specific journal,
Okay.
and the number of citations on my paper, how important or how much value does that carry?
Yeah, that of course that carries value, but it takes time to generate the citation, and it carries value, meaning it's because let's say I'm starting some new project, so I'll go by good papers, like how many citation it has, unless I'm extremely desperate, that then I'll go whatever is published and I will try to add that. But good citation, it adds value, it adds authenticity. It says like there are other people who are agreeing to that and using that that data. Does that answer you?
Yes,
I
ma'am. Thank you.
can I say
Yeah.
something?
Yeah, go ahead.
So
Yes, sir.
it definitely, I'm not sure if what I will say is relevant or not, but this is definitely going back to the debate of journals with high impact factor versus not so high impact factor. Like this is journals, they are like, when you go to a mall, they are like those clothing store, Gap, Old Navy, right? So you definitely trust, like, all the clothing that they sell. This is like, sort of reading papers from, like, those high impact journals. But like, let's say you can also definitely find good clothing in not so famous like, store, like equally good. They're definitely cheap because they don't have a high brand, but like, quality wise, they are the same material, right? So those are basically your journals with less visibility or relatively not so high impact factor. And people, journals with high impact factor, just because they are now famous, they have more credibility, they do definitely, they do thorough investigation of your work. And because of that, publishing in those journals, that take time, long time, long time. Sometimes 2 years, sometimes, like, this is beyond imagination. So people, like, let's say, who are in a hurry to publish something. So like, let's say, but if you don't publish now, someone else might publish it. There are always those sort of competition out there. So you don't want to, like, submit to Nature Cell Science, because that they will take year and a half just to get back to you. And by the time someone else will publish the data, so you rather, like, take the loss and go to a second tier journals. Well, second tier means still pretty good, like, not like, yeah, like, the next level journal. And then so that where they don't take 1.5 years to get back to you, they take a week to get back to you. So these are also parameters that you have to consider when you submit your work. Sometimes, like, your work is really good, is unique, but when you submit to a journal, they take time, right? And during that time, something like that comes out from some other group. So of course they can no longer accept your work, so they reject your work. Then the impact factor of the journal where you finally submit to it go down so much, like, drastically it goes down. So there are all those complicated things. There is politics everywhere, so
Yeah, I want to add one more thing. I forgot to mention about peer reviewed. The authenticity, like how many citation you got. Then another authenticity is peer reviewed. So when you do your research, when you try to find thousands and thousands of literature, you are trying to pull whichever is authentic, go for those peer reviewed. Those are your top tier one. Peer reviewed meaning this is accepted, almost accepted and all. And again, there will be your colleagues, like whoever are in that arena doing this kind of research, or they have some sort of understanding. They are going to review your literature, and they are going to post their comments and what they think, and what are the scope of some improvement and all. If those papers are there, pick those. Peer reviewed are more authentic and more scrutinized. Anybody else has any other question?
Thank you,
Hi
ma'am.
everyone. Hi everyone. This is Kiran. I have a question. Do we get a chance to work on the paper during this course?
Meaning?
Will we be conducting any active research?
Oh,
during this
that's
famous course?
Dr. Bal. Dr. Bal has some small stuff. I know it's impossible to do it over Zoom and all, but he has some small stuff, and he has even a setup in Nimapara, Odisha. But what we are trying to do is just to give you the gist of what he will do. He will try to conduct some small stuff, like some sort of review, so that you can have your names in one of the articles. So he will explain more. Dr. Bal, are you there? You are mute. Can you unmute yourself, Dr. Subhasis Bal? Probably internet connection, I think. Okay, so what he was planning was to involve you all in those small reviewed articles, and show you some small experiments. But his expertise is in agro research. But right now he was trying to collaborate with one of the group of researchers who are conducting research on gut bacteria and COVID transformation. So he will be able to explain that to you later. I mean, he will conduct few sessions too. For now, this is all I can say. Looks like he is disconnected, or his internet not working. I'm not sure. Okay, next.
Ma'am, one more question, if you don't
Yeah.
mind.
Yeah.
At the end of the course, we will get a certificate, right?
Yeah.
Yeah.
For that, you have
which,
to. Yeah. Tell me.
Yeah, which letterhead will it be, ma'am?
Letterhead meaning?
On the like from which like what will be the?
That's the Beautiful Mind Youth Mentoring USA.
Okay.
You're gonna get it from us.
Okay. Thank you.
Mhm. Anybody else?
Hello.
Dr. We are missing you. Actually, somebody has a question for you.
No, I'm here only, but I got some problem in that unmute. I was unable,
Okay.
unable to unmute
I
that.
can understand. Okay, so you have the floor. let's wrap it up as soon as possible because it's already getting over time. I mean, I already informed them you're going to conduct other sessions and all. So they wanted to know if there are some way they can have their name published for small stuff, or they get to see some hands-on experience and all. So please go ahead and explain. What are your plan for later?
Yes, madam. Thank you everyone for joining us today. And I don't know, different part of the globe people joined with you. So that maybe good morning and good evening for us. So myself, Dr. Subhasis from Bhubaneswar, Odisha. And my educational background is I am from Utkal University, MSc and PhD from Utkal University, Bhubaneswar, Odisha, India. And then I did the postdoc at Institute of Minerals and Metals Technology, Bhubaneswar, Odisha, for some years. Then joined as a postdoc at RPSC, Regional Plant Research Center, Bhubaneswar. Then as a postdoc researcher at NRCPB, National Research Center on Plant Biotechnology, New Delhi, India. Then Indian Institute of Rice Research, Hyderabad, India. Then as a postdoc scientist at Indian Institute of Oilseed Research, Hyderabad, India. Then as a postdoc researcher at the Department of Agriculture Science, Mauritius, University of Mauritius. And presently I am working as a national consultant for regenerating farming, ERI, for India office, International Rice Research Institute for India office. And I have my own farm, Surya Seeds Research, where I am conducting research on regenerative farming, natural farming, and organic farming, and precision farming. 4 or 5 types of farming models are there. So I am working on that. And some college students are also participated in that experiment with me, so that they can get some publication for their career enhancement. So Dr. Parasar already told you how to publish a paper in a good journal. So publication is most important for all researchers to get a good job, to get a recognition around the globe, and to validate their research work around the globe, so that people can understand what is going on every aspects like agriculture, animal science, medicine, and other part of the research area. So it is a updated format. Whatever you are doing, it will reach throughout the globe, so that people can get benefit from it. So I am not going that much to the research part. Already Dr. Parasar explained you regarding the research article. So I am working on reverse part. So I right now I close all the publications for sometimes, not for the future. For sometimes I close my publication, I stop my publication. So I don't mind. The exact true thing I want to share. 90%, 95% people around the globe publishing paper to get a job, to fame their name. And apart from that, 5 to 10% people are real researchers. Those publishing paper to get a job and to help the society in different aspects, so that their research work can reach every part of the globe, so that we can save the globes in terms of ecosystem, biodiversity, agriculture field, all aspects of research field we can save through a good publication. So I'm not going through that so much. Madam, can you please share my slides from the beginning? 3 slides I shared to you on my work.
No, it's you said it's so late. I won't be able to download it to the cloud. So it takes time. So next time,
Acha. Okay.
so it doesn't happen that.
May I know the participants are from which background? Just within 1 minute you can complete. 15 or 17, I think the 14 participants are there.
There are many, most of them are physicians. Either they're practicing or they're studying.
Oh, from medical background.
Yeah.
Oh, it will be a little bit difficult for me to convince them on agriculture. Okay, I'll try my best. Madam, up to 9:30 I can present. After 9:30 I have a presentation for IRI, International Rice Research Institute. 15 minutes I have time right now today.
But take less. Actually, we will assign you other workshops. Take like way less. Maybe 5 minutes if you can wrap it up.
Yes.
Okay, thank you.
Dear past participants, today we have very less time for me. That's why I am sorry for that. All of you are so much interested for a publication as you are all you are researcher and it needs for your career development. So I am basically a agriculture background student and agriculture background scientist. So in the near future, we can plan how to produce azolla. Azolla is small form which provide natural nitrogen to the plant, to the rice plant. Basically, I'm a rice scientist. So how it can give natural nitrogen to the rice. Then we can design vermicompost unit. Then we can design format manure composting unit, cow dung unit, biogas unit. How we can regenerate topsoils and how we can do a biochar unit, a green manure unit. How can we reuse the kitchen wastes? So like that 40 to 50 small experiments are there. So basically on the first of the work, we can do literature survey. How many, if I am talking about azolla, we'll go through that. How many countries involved in azolla production? Just a review paper. We'll go through first. We'll go through the review paper. So how many papers till now publishing on the azolla, so that we can get updated knowledge around the globe on azolla production and its use for the environment and for the crop. So in the same way, for every small experiment, we can publish a review paper. So already Dr. Parasar told how to write a review paper. Because if you have the facility, you can do a small experiment, and whatever your data you are getting, you can compile it and you can publish in small journal first. If you have good data, you can go for a higher impact factor journal. That is depend upon you, how much time you have and how much facility you have to produce or to generate the data. So for that, we have near about 40 to 50 small experiments in the initial phase. So you just talk with madam. Then in the next session, I can arrange individually, or we can arrange groups like 4 to 5 candidates will be there. And in collaboration, we can publish some papers in small journals first. So right.
We will be together, and actually after this, how many kids are there and all, then we can make them as based on their interest. We can divide them in different groups and assign them work.
Yes, madam. Just I want to explain nowadays what people around the globe facing. So today we are eating just poison. With every meal we are taking minimum 10 mg of poison. It is coming from agriculture field only, through food, through snacks, through different kind of things. So that is a big problem for everyone. We are not bothering what we are eating in the table. So we are just running behind sports, cinemas, movies, all these things. But whatever coming from the agriculture field, we don't know things. So, now recently, people from last 2 years, people faced a different kind of problems like high blood cancer, high blood pressure, high and low blood pressure, type 2 diabetes, type 1 and type 2 diabetes, and gastric problem, and obesity, and these are 4 or 5 important diseases around the globe, people suffering. More than, if we'll count, more than those are crossing more than 50-year age, we'll get 70% people are suffering from these 5 different kind of diseases. So for that, I have developed a model, so through which we can produce so at least residue-free or chemical-free food. So it is my experiment right now I'm doing in my own field. So that when time will come, we'll do small experiments, particular on organic farming and regenerative farming, how we can reduce that poisons from our food. So today, little bit time, I'll be I can share 5 minutes time. So because I have to prepare for that next my ERI meeting. So without slide, it is very difficult because I have already prepared. I'll go through the slides.
That's okay.
It will
It's an introductory session, so
Yes,
at least
yes.
we get to tell everybody what we are about to do in those next sessions and all. So based on their interest, we can expand. We can expand to different groups. We can expand to different projects. So let's first say how. I mean, I was interested. I was curious to see how this works out for today. Then we will expand and we will do more stuff. Okay. So
Yes,
thank you so much, Dr. Bal. That's actually interesting. But in a way, if you can find to incorporate more, like you have to a breeze between biology and, I mean, zoology and botany. So plant and animal. So we
yes,
can expand more on that, and
yes,
everybody can be benefited. Okay.
yes, madam.
Okay. Thank you so much. Dr. Saptadipa, are you here?
Yes, ma'am.
Okay, so I would like to conclude the meeting. So please introduce yourself, and she's the woman behind all of this. She's working really hard in collaborating and in conducting those small talk in between all those participants. So please go ahead.
Hello everyone. I'm Dr. Saptadipa, a attending pediatrician from Delhi government. I'm actually more into the clinical side, clinical treatment side. But nowadays, fortunately, I am having a great friendship with a guide like Suchispita ma'am and Dr. Parashar. So I'm really interested for clinical research in future also. Nowadays involved in few projects. So actually, I feel like a research experience makes a huge difference in your resume, especially if you're a USMLE candidate, because USA is something. It's a place which is far ahead of us in all the active research areas. So good luck. I hope these upcoming sessions are going to be helpful for you all. And definitely at the end of 3 month session, you are going to get official certificates so that you can show that for your resume. And it's not about only resume. This is going to help you to enhance your knowledge also, because you all are most probably MBBS students and very less knowledge about the active research part. As far as I know, I have already completed residency here. Most of the time, students submit the thesis for the sake of exam or for the sake of publications, but not for
Exactly.
active research part. So here you are going to learn the basics: how to write research paper, how to know what are the research methodologies, what are the articles, how to read active journals, papers. So I think this is more about gaining knowledge than only the resume thing. But ultimately, we all are preparing for residency in USA students. We are competing with USA students. So I think this project is going to helpful for your future and for your resume also. Thank you. Good luck.
Can I ask a small doubt, please?
Yes, yes.
I just wanted to know the schedule of the upcoming sessions and how long it's going to last.
I will.
And what will we be discussing in those sessions?
We will send it to you via email. Okay, with the roster and everything. Okay.
Thank you. Yeah.
Thank you. Okay, so that's it. We are concluding now. I guess you don't have any other question. If you are coming up with any other doubts or anything, email us. You have our email ID. Then we will inform you about our future sessions and their dates and what you are planning to do and how we are segregating those groups and all. So that's it for today. Thank you so much for attending.
Thank you.
Yeah.
The session is really informative. I mean, it's it has covered pretty good information for an informative session. And I'm really hoping this would benefit all of us a lot. And thanks a lot for organizing this event.
Thank you, Vaishnavi. Thank you. Thank you so much.
Thank you all.